Welcome to our university, the Central Luzon State University, a 658-hectare campus in Science City of Muñoz, Nueva Ecija. It is the lead agency of the Muñoz Science Community and the seat of the Regional Research and Development Center in Central Luzon. The Biotechnology and Analytical Laboratory is about 1 kilometer from the CLSU main gate. This is where our laboratory is located. It promotes the use of biotechnology in agriculture, applying the molecular markers, genetic engineering, molecular diagnostics for animal and plant diseases, plant tissue, cell culture, and micropropagation techniques. Let's go inside and I'll show you around. We need to remove our outdoor shoes and wear these lab slippers, okay? Let's go inside. This laboratory represents Biosafety Level 1 and nearly Biosafety Level 2. With both Biosafety Cabinet Levels 1 and 2. This is a laminar flow hood. Biosafety Cabinet might be confused with the laminar hood as both of these pieces of equipment work as enclosed workspaces. This only provides protection to the sample and not to the personnel. Nucleic acid extraction methods are the keys to molecular biology. If you're going to analyze any DNA from human, animals, plants, microorganisms, viruses, and other organisms with DNA or RNA, you need to extract your DNA or RNA before doing your PCR or LAMP. Other labs use commercially made extraction kits like this. But in our project, we are attempting to design an improved LAMP assay kits for field-based diagnostics for Schistosoma japonicum and leptospirosis. In this, nucleic acid extraction and LAMP assay are included in one that can be applied away from the laboratory. This extraction procedure does not need long methods, but the use of the kits that will be created will be imparted to you. One of the instruments we use in nucleic acid extraction is the microcentrifuge. A microcentrifuge, also called a microfuge, is used to spin small tubes with 2 ml or less liquid samples at high speeds, generally up to 15,000 rpm. Centrifugation is important for many biological applications such as pelleting nucleic acids or proteins from solution so that you can collect your DNA at the bottom, or simply to gather those last precious drops of liquid into the bottom of the tube like this smallest mini spinner. The vortex mixer is a vital piece of equipment commonly found in research laboratories to mix samples in test tubes, well plates, or flasks. And of course, this is our ultimate weapon in molecular laboratory, the micropipette. The micropipette is used to transfer small amounts less than 1 ml of liquids, usually down to 0.1 microliter. The scales on the micropipettes are in microliters, and 1 milliliter is equal to 1,000 microliters. All laboratories have huge storage of biological samples and different solutions that require proper storage. Failing to store the samples at their given temperatures can result in inaccurate results. Proper storage practices are extremely important for any laboratory. Biological reagents and buffers that are used to daily basis should be kept in refrigerators. Some common examples include enzymes, buffers, and cell culture media. A molecular laboratory would not be complete without these, the thermal cyclers or thermocyclers. They are sometimes called molecular copier, using the technique called the polymerase chain reaction or PCR. Significant amounts of a sample of DNA are necessary for molecular and genetic analysis. Studies of isolated pieces of DNA are nearly impossible without PCR amplification. What's really important in doing PCR is your DNA of interest, of your target parasites, microorganisms, viruses, and even plants or animals or human. The thermocycler raises and lowers the temperature of the samples in a holding block like this in discrete, pre-programmed steps. The lid is usually heated to prevent evaporation from your reaction. Inside are the wells where you put in your 0.2 ml tubes. Usually this block can hold 96 tubes or 96 reaction samples for analysis. For example, in testing COVID, one tube reaction represents a test for one patient. The results of a PCR reaction are usually visualized or made visible using gel electrophoresis. Gel electrophoresis is a technique in which fragments of DNA are pulled through a gel matrix by an electric current, and it separates DNA fragments according to size. A standard or DNA ladder is typically included so that the size of the fragments of the PCR sample can be determined. DNA fragments of the same length form a band on the gel which can be seen by our naked eye if the gel is stained with DNA binding dye. Here is an example of the sizer ladder, which is like a ruler, so that you will note how many base pairs that your PCR products have. After the DNA has migrated through the gel, it needs to be visualized so we can determine the size of the DNA molecules in the sample. Since DNA is not visible to the naked eye, we stain it with a fluorescent dye. Fluorescent dyes give much better detection levels when imaging the gels. The next step is to capture an image in a gel documentation system. Gel documentation system, or gel docs for short, use high-sensitivity cameras to capture images of the gel or the agarose gels. Often, these systems are equipped with UV or blue light transilluminators, which are used to excite the fluorescent stains, which then emit light which can be captured by the camera. Once you've captured an image, you can analyze it based on the pattern of the DNA bands to determine their size and the quantity of DNA present. You will know that your sample is positive if you see the bands and got the correct size. Now, these are the basic procedures and instruments that a molecular laboratory are usually performing. Please stay tuned for the next video on how we perform the loop-mediated isothermal amplification, or the LAMP. See you!